Hello, Year 12s. Welcome to my first podcast. Not podcast, what's it called? Screencast. Um, yeah, it's probably going to be a bit of a disaster, but just bear with. Let me know if these are useful or not, because I will stop doing them if they're not. I just figured that as well as giving you the slides to make notes on and read and things like that, you might benefit from me talking through it. And sometimes I think of things on the spot when I'm talking that I might not have put in the slides. So that's why I'm doing this. We are running a little bit behind schedule. We really should have covered active transport last term, but we didn't get around to it. We still need to do respiration, photosynthesis, a bunch of other cell processes as part of this external. So we really need to finish active transport in the next couple of days. Um, yeah, there's heaps of stuff to cover, so I really recommend that you take responsibility, make some notes, be autonomous about your learning because I can't do much in my position at the moment. And yeah, we won't be touching on this stuff until the end of the year, and it's essential that you understand it for the external. So please take it upon yourselves to do the tasks that I set, make yourselves some notes, email me anytime with any questions. Um, yeah, you're seniors now, so make me proud. The first part of this set of slides, I've basically put together some recappy stuff about the beginning of this unit. So all the stuff about cells, specialized cells, the plasma membrane, which is essential in your understanding for this unit. So recognizing the fluid mosaic model is made up of phospholipids, proteins, carbohydrates, cholesterol, all of that kind of good stuff, and why it's there, how it works, um, what's involved with passive and active transport, etc. So I've recapped all that stuff in the slideshow because I'm a nerd and I'm trying to be helpful. And then I've also put a recap of passive transport, which we've already covered, which is essentially the movement of any substance from high to low concentration, which means down its concentration gradient. And because it's going downhill, it doesn't require any energy. It just does this naturally, randomly, using kinetic energy of molecules that they all have, such as in these GIFs here. Okay, three types of passive transport. We looked at very simple diffusion, which is just the movement of particles by slipping through the phospholipids in the membrane. Facilitated diffusion, which is the movement through the membrane from high to low. However, if the molecules are too big or too charged and they can't just slip through the phospholipids, they use protein channels, which are just kind of big tubes that don't have to expend any energy to let anything through. And osmosis, which is the diffusion of water. Okay, you need to be able to use your tonicities, your relative hypertonic, isotonic, all of these keywords that we've been practicing for those three. So once you're quite confident with passive transport, which you should be by now, as you can see, I've hyperlinked a bunch of stuff into these slides, so you can go back and look at them, but they're all on Classroom as well. Then we need to move on to active transport. So we've covered yellow, which is the part of cell transport to do with going from high to low concentration, which does not require expenditure of ATP or cellular energy. Now we need to do active transport over here, which is the opposite, moving from low concentration to high, so pushing things into an area that already has a lot of them, which is going to require the cell to give up some of its energy that it has created. Oh, that's a typo, whoopsies. Cool, active transport in cells. So you should be able to, by the end of this lesson, describe the process of active transport, understand how and why it is different to passive transport. So comparing and contrasting these two terms is really important understanding, kind of abstract understanding that you need to be able to apply to exam questions. Bum, bum, bum. So what if instead of spreading out via diffusion, the cell wants a certain substance to become more and more concentrated inside the cell? So it will have to force lots and lots of stuff into the cell, or in reverse, it might want to get rid of a lot of stuff, moving it outside the cell until it has very little. It's going to have to use active transport, which is the process of moving something from an area where it is at a low concentration into an area where there is already a lot of it. This requires the use of cellular energy, or ATP. So you can use these two terms relatively interchangeably. ATP, or adenosine triphosphate, is a molecule, and it releases cellular energy. So either one of those will do. I suggest writing these kinds of things down and yeah, making yourselves notes as we would in class because it's really important stuff and it just doesn't sink in if you're not writing it down, in my opinion. So please do that. So we're going to look at two different processes that require energy and are different forms of active transport. The first one looks very similar to facilitated diffusion, so it requires the use of a protein in the membrane. However, again, we can see here that the substance is moving from the area where there is not a lot of it, low concentration, to the area that it is in high concentration. So this requires the protein to pump it through actively, really force it through the membrane. And that's what requires ATP. So in these cases, we call this protein a protein pump. It's thinking of the word pump as requiring force, whereas in the other case, it was protein carriers or channels that were just kind of big hollow tubes that could allow things to diffuse naturally without using up cellular energy. So it does look similar to facilitated diffusion, but it is different in that we're going low to high and using up cellular energy. The second type that we'll cover next is called cytosis, and it is the act of the cell basically eating or drinking very large quantities or large molecules of a substance. Notice that the molecules of the green substance are being actively pulled through the membrane from low to high concentration. Okay, so the area on the right, or inside the cell in this case, is becoming more and more concentrated as the transport carries on. I'm just skimming these. You can pause or you can just look at the slides later, so I'm going to upload them separately, but I'll just say things that I think are important. So this is just talking about the protein again. So you might be asking, you probably aren't, but you might be, why a cell would want to build up a high level of something inside of itself or get rid of so much of itself that it's making a very high concentration outside the cell. So why would it want to do that? There's a few examples that we can use that might make you go, that makes sense. And sometimes these are important to be able to really draw home your point in an exam question or apply it to a situation. So the first one would be the use of active transport in very specialized cells in our bodies called neurons or nerve cells. So as you can see, this cell here does not look like our typical drawing of an animal cell. This part kind of does, but the cell itself has all these dendrites and an axon and all these kind of funky things going on, and that's because it is very specialized. So we've talked about specialized cells, and this one is specialized in that it has a lot of what we call sodium potassium pumps along its axon, this big long skinny thing here, and they're used for really the only point of a nerve, which is to communicate with other nerves all up and down the body to make you do the things that you do, whether it's conscious things like moving your muscles, being active, or things that you don't even control, like the walls of your intestines moving to digest food, or your heart beating, the muscles in your eye expanding and contracting your pupil when it's light outside. All of these things are controlled by neurons. And a massive part in how they work is the use of active transport in and out of the membrane along this axon here. So a nerve impulse or action potential is simply a flip in relative charge between the inside and the outside of the membrane of a nerve cell. And it travels along like a chain reaction until it reaches the end, in which this end of the cell will communicate with the next neuron and so on until the message gets where it needs to go. So what happens is quite simple. Potassium and sodium are both small charged ions, which is an atom that's either lost or gained an electron. In this case, they're both positive. Apologies for this mistake. Both positive, they've both lost an electron, and they can simply diffuse through the membrane by themselves. However, the cell doesn't want that to happen. It wants to maintain a potential difference in charge on either side of the membrane so that it can send these impulses when it wants to. So what it has to do is pump ions against where they're trying to go naturally through passive diffusion or simple diffusion, using this protein pump in the membrane. And this is going to require energy. So the cell wants to maintain a high level of potassium inside and a low concentration relative to the outside of sodium. So it's going to be moving these ions against their concentration gradient. And how this happens is, if you look down at this GIF here, sodium will move into this pump and a molecule of ATP will snap. And this snap is what releases heaps of energy. And that extra phosphate group that's snapped off of ATP is able to bind with the protein pump, which changes the shape of the pump, allowing it to push usually about 3 ions of sodium outside the cell, so pushing it into the extracellular space and keeping that at a high concentration. When that phosphate group releases itself from the pump, just trying to make sure I don't use any of the wrong words, it allows for the movement of usually 2 ions of potassium into the cell, keeping that concentration high inside the cell. So both of these movements, pushing sodium out, pulling sodium in, require a change in shape of the protein pump, and this is facilitated by the release of energy from a molecule of ATP when it snaps into a molecule of ADP, diphosphate, and a phosphate group, which is used when it binds to the protein as it helps it to change shape. I hope I said that right. That was a lot of blurred words. So it's not essential that you understand the exact mechanism behind this, except for the fact that the cell has to use up a molecule of energy, or ATP, in order to move sodium, potassium across the membrane. You might have noticed that both of these ions are positively charged, except for my inaccurate title up here. So the way that it's going to change the relative charge across the membrane is using different amounts. So if equal amounts of Na and K were transported across the membrane by the pump, the net charge transfer would be 0, yeah, because they're both positive 1. There'd be no net flow of current and no effect on the membrane potential, which is not going to help at all. So most neurons will be moving 3 sodium ions for every 1 potassium or 2 potassium ions. And that keeps a potential difference across the membrane maintained. And then it can send its nerve impulses as it needs to. What's next? Yep, so that's just what I said about ATP. If you want to know exactly how it works, that's a molecule of ATP, or more specifically, it looks like this. And when that phosphate group snaps off, that is what releases the energy. Okay, and it's this phosphate group here which is able to bind to the pump and actually make it change shape and pump things against their gradients. Okay, I couldn't help myself. I found this on Google and it was crack up but super lame. Please forgive me. But also, who makes this? Another example, so example number 2, root hair cells. So we've probably learnt about root hair cells a number of times. They're a specialized cell found in the roots of plants. They have this very extreme extension out the side, which increases the surface area of that cell. Okay, so think of heaps and heaps and heaps of these cells making up the roots of a plant to really increase the surface area for absorbing minerals and water and stuff like that from the soil. The plant needs to be able to get a lot of minerals into the root hair cells to then transport them to other places in the plant. So for a short time, these root hair cells are going to have to be really high in concentration because they want to be able to send a lot out to the plant. However, once they get to a certain concentration that is isotonic to the soil, diffusion would cease to happen. There would be equal flow of minerals into and out of the root hair cell. So the plant is going to need to use active transport to make sure that it keeps pulling minerals into the root hair cells even when they get to a really high concentration, because it needs a lot of these ions and minerals and things like that to send to the rest of the plant. So they have special pumps in their membranes for moving essential minerals into the plant against the concentration gradient. So as you can see in this picture, this vacuole in the root hair cell is full of, let's say, magnesium, for example, or nitrates, but it wants to keep pulling more in, so it's going to need to use a pump to move them from where they're in low concentration outside the cell to high concentration up their concentration gradient. Okay, I'm going to have to stop there because I'm about to run out of time. See you in a minute.